So like I said, the theme for today is young scientists, or at least scientists younger than I am, with big brains either in their heads or in their heads and as study objects. And Gary Motz continues that theme. He is currently finishing his PhD at the University of Cincinnati, where he is a paleontologist. He is a paleontologist here and involved in the Center for Biological Research Collections. I'm rather envious. He is smart enough to be a paleontologist. I am not. And yet he chooses to study invertebrates rather than backbones and brains and that sort of thing. So Gary's really, he's an exceptional talent in really in informatics and information technology, as well as paleontology. Done some very, very striking and very impressive work. And in fact has participated in the development of work that we have sitting, awaiting future funding, which I expect. I'm just sitting by the post office box waiting for the check to arrive to continue some of the work that we've been involved in. And it really is a pleasure to introduce Gary as the second of our Peebles Lecture Series speakers this morning. And take it away.
Thank you, sir. Well, good morning, and thanks everybody for coming. I'm going to focus a little bit on some more broad synergies in this sort of big data ecosystem that we have here, and that it really is this environment of research technology and the synergies between all of these functional units that allow for research data to really come to a new birth in the digital age, and then metamorphose into a fuller, richer ecosystem environment for understanding and conceptualizing science and nature and our understanding of this universe, but particularly from a computer science standpoint. And so I'm going to be talking a lot about how this technology is pervasive and how that pervasiveness allows for us to understand a whole lot more when you start to attack a bunch of different questions from lots of different angles and approaches. A number of years ago, and this is sort of one of the big things that has caught my eye as I was applying for this job here at IU, and why I sort of ditched out on finishing my PhD early, is I realized that this was an opportunity that was not worth missing. I wanted to sort of throw myself into the additional challenge of working a more than full-time job that I love here at IU, and also work on my dissertation simultaneously. So call me a glutton for punishment. But this digitization of media and about all the collections resources that we have here at Indiana University, which are tremendous and really quite rich, is something that we're only starting to tap into the real resources and the products from these initiatives. And so as we're digitizing these research collections, whether they be with MDPI, or with what the Center for Biological Research Collections has me doing with biology and geology and anthropology, it really is a concerted effort to try and make sure that this dark data can come to light. We have a very rich tradition of cyberinfrastructure and informatics here at IU, but the natural history collections universe has extended considerably further back at this university. And so the Center for Biological Research Collections exists as a sort of a temporary entity that enhances collections-based research that really focuses on driving those synergies and the shared infrastructural support for biodiversity, informatics, and for collections-based research. And so we are sort of this central hub of organization for policy and for education, outreach, and research, and the way that each of these spokes of the wheel come together to provide this shared platform for biological research. And so as I was saying about the history of all of this, and so we've got three different major collections that go and feed into this understanding of the natural history of Indiana and more broadly of our natural world. Each of these collections were founded in three different departments across the College of Arts and Sciences, and the Indiana University Herbarium was founded in 1885 by a botanist, an amateur collector, and I was actually no amateur, he was more an avocational collector, who really was told by his doctor that he needed to find a hobby. And he was a pharmacist by training, and he decided to go pick up plants for his stress reliever. So he went systematically and collected the vascular plants of Indiana on a county-by-county scale. And so because of that stress relief practice that was encouraged by medical practitioners of the day, we now have a very, very rich information system that is geospatially referenced across the entirety of the state, but also can serve as this backbone for DNA studies and for genetic information sciences and for other practices that were not necessarily considered viable at the time, nor are we even thinking about today. Similarly, for the IU Paleontology Collection, where it was formally established in 1903, the collections that we have in the IU Paleontology Collection extend far back beyond that, back to the early days of Lewis and Clark, when congressionally sanctioned expeditions were coming into Indiana. And so there's a large statue right outside of Alumni Hall in the Indiana Memorial Union of the very first state geologist of Indiana, whose family members were critical to establishing this university and to the paleontology collection here at IU, which now has a collection of roughly 1.5 million fossil specimens, but those are technically collection lots, which can be a box, and so the actual number of fossils we have in our collection is easily above 5 million. And the Zooarchaeology Laboratory, which was in the anthropology department, which has roughly 11,000 vertebrate skeletons. So the number of elements and moving parts that go into this sort of biodiversity infrastructure really are quite tangible in the physical sense that we have the bones and the fossils and the pressed plants that are glued down onto pieces of paper. But really the information that they provide is far, far more rich than just that. So just to give you a brief walkthrough of sort of our digitization protocols and how we're trying to bring these dark data to light, the Herbarium has a robust organizational system that is all of these curated plants are pressed on pieces of paper and then categorized by folder that are sorted taxonomically. So the entire Herbarium of collection of flattened sheets are organized in a systematic sense, so that way you can go and find those that are most closely related to it in the same cabinet or in the same folder. And then we've also got them sort of geographically. So the entire collection itself is a giant filing system for organization for big data practices. So as we're systematically going through and using a custom-built diffuse light box for photographing these images, we slide the images into there, we get some very high resolution imagery of very beautiful collection of plants and from all over the state of Indiana and more recently beyond. And so as is best botanical practice, the best information that we have about all these collections are not only contained within the field notebooks that are associated with these collections, but also printed in the bottom right-hand corner of each of those sheets. So there is provenance information about who collected that plant, from exactly where they collected that plant, information about the taxonomic determination and what species that belongs to, in addition to other information like associated species and habitat and other things like that. So as I mentioned, this is something that we can put together in this sort of big data context and sort of put together a historical and ecological baseline for understanding sort of the geospatial information system that goes into our understanding of what the plant life and the foliage of and flora of Indiana looked like prior to civilization coming into the state, or looking, because we do have some very historic collections that occurred prior to the deforestation of the state of Indiana. And so we've got for grand challenges and for understanding sort of how the history of life has evolved across the state of Indiana especially, we have a very good ecological baseline in the Charles D. Herbarium. The Zooarchaeology Lab is similarly focused on the Midwest, but has a number of collections from across the Great Plains of the United States, and is a really, really wonderful resource in that we have a dedicated graduate student to help corral volunteers that come in, because usually there are 20 or more student volunteers that come in and dedicate at least 10 hours a week to helping to curate this collection. And so this is something where they are a very active community of students that are interested in not only getting their hands on the bones and understanding, well, how do I identify this bone, or if I found this thing in my backyard, how do I figure out what this is? I'm not going to go look through some really dense technical jargon in a book. I want something to put my hands on and say, oh yeah, this looks more like this one instead of instead of this one. So having physical reference collections like this one, where you have comparative faunal osteology, allows for us to go back to the reference set and say that this is what this is versus what this is, or also putting together similar ecological parameters that allow us to understand what the fauna of the Great Plains and Midwest looked like 200 years ago or more. Because some of these, as the name of the laboratory indicates, are archaeological finds. And so many of the collections here, I'm sure that Dr. Peebles was very much involved with in the early days, and a lot of these actually are from Angel Mound State Historic Site down in southern Indiana. So in the digitization of this lab, we do have some sort of paper-based media and metadata that goes along with this, but the vast majority of them have been are these bones, and as three-dimensional collection objects, we've been digitizing them with a number of different methods that involve either very, very high resolution 3D laser scanning or structured light scanning that we've entered into partnership with the Advanced Visualization Lab division of RT to really put together this framework for digitizing all sorts of bones and other artifacts from both zooarchaeology and paleontology, where they're not just conveniently flattened onto an 11 by 17 piece of paper, where there's a lot more textural information that when you map the color information onto the bone itself, or you remove that information, you can start to see the actual suturing of the epiphyseal plates and looking at how the growth of that individual organism was determined and whether or not it's a juvenile organism or was a more adult, or there's lots more that you can tell by the different ways in which you can visualize that those specimens. And so as we've been working to digitize these collections, we've got anything that range from the American bison to a pronghorn antelope up in the top right-hand corner. This is a I call it a filleted gecko, but it's more of a traditional Chinese. It is in fact a gecko, but it's on a popsicle stick, and its sort of sagittal sections have been pulled apart to make as a medicinal herb, and so it's intended to be used in either a soup or a tea for ailments in traditional Chinese medicine. And so we've created some 3D models of that, and also the image on the bottom left there is one that was it's a piece of pottery from the Quapaw tribe, and there's a tribe that Indiana University has developed a very good working relationship with to determine how can we best work with the first Americans and the first people that lived in this area and respect their traditions and their culture by holding on to the things that we've gone and collected from their heritage areas and from the historic sites now, but also to return those back to the people from whom they belong and are that cultural heritage. So the Quapaw tribe has allowed for us to digitize some of their collections to retain them as research objects for us to continue to utilize, but then we return those and repatriate those back to their formal owners. And I mentioned a little bit about the volunteer engagement in the student collection in the Zooarchaeology Lab. This is one thing that we've developed as a tradition over the past several years, is to have a bone of the week on the lab's Facebook page. And so the students will frequently sort of race to see who can find the answer to the bone of the week, and they'll just put a picture up of a bone from the collection and see if you can figure out what animal it's from and what age the animal is, or what type of bone it is. But even to a formally trained faunal osteologist, which I am not, these bones look strikingly similar. And when you put these into 3D models and are able to spin them around and embed this Sketchfab link onto Facebook, you've now got a 3D bone of the week that allows a little bit more closer inspection where you can zoom in, you can pan to each of the different features that can really begin to differentiate what each of these four bones are. So if we've got a bighorn sheep, a mountain goat, a pronghorn antelope, and a white-tailed deer all in this one image, can you tell the difference? If you had them in your hand, the physical bones themselves, or a 3D print of those bones, would you be able to tell the difference? So divorcing these collections from their context renders them essentially useless, but at the same time it is that rich contextual metadata that allows us to differentiate each of these. And so I know that you guys will probably be asking me about this later. The bighorn sheep is specimen D. C is the pronghorn antelope. B is the mountain goat. And A is the white-tailed deer. One of the other things that's interesting, this is shared publicly on our Sketchpad page, is that specimen A there, and PowerPoint hasn't quite figured out how to embed 3D models just yet, is if you were to zoom in and manipulate this, you can see some butchering scars on here. So we're actually, the greater trochanter of this femur, the upper limb bone of that white-tailed deer bone, has been butchered and presumably consumed by Native American people. So that is an archaeological specimen from our collection that we know has a very distinct tie to human cultural interaction. Also, we do a little bit of forensic reconstruction where you take modeling clay and were to recreate the face and put the muscular structure onto some human remains in order to determine how we can best put a story to the history and the life of a particular individual. Whereas you can't do that, especially with Native American skull, especially one that's been so highly fragmented and has such a storied history as this individual does. But 3D printing has allowed for us to then 3D print the models and then ship that 3D print off to a colleague at a medical hospital in Boston for them to do the forensic reconstruction of a pretty relatively inexpensive 3D print job, rather than sending a priceless artifact through the mail or hand carrying it up to Boston. It's all kinds of cultural and logistical issues that preclude that from happening. And so all of these things that are locked away in the collections and that are dark data, unless you're a scientist that actually specifically works with that particular collection, really helps us to make sure that these data are shared and appreciated and respected for the traditions that they enable us to know about, but also to broaden their exposure and experience to people from beyond just the academic community that's specifically working with them. And so, sort of as a tribute to, as this is the Peebles Memorial Lecture, this is from the Angel Mounds State Historic Site down in southern Indiana. So this is a historic burial of a common dog, but this dog was not quite so common as you'd think. For the context and the cultural period of the time, dogs were not ceremonially buried or were not generally laid out in sort of a resting position. They are sort of, and there are a couple other hits within this photograph where there are others dogs buried sort of in the same area in the same context, but they're just piles of bones that are just done and buried without much thought to them. But with this particular instance, there is not only the sort of a resting pose for this particular dog, which is substantially larger than the average dog of any the domesticated animal at the time. It's also buried with some other ceremonial type bones, which are golden eagle or red-tailed hawk and other bones that connote some significance to the story of this particular animal. And so we can start to put together the context of this burial and do a virtual reconstruction not only of the bones themselves, but of the entire framework, and start to help understand the greater context in which all of these show a much larger picture. One of the biggest projects that I've been working on lately, especially with the Advanced Visualization Lab, is to try and work on photogrammetry and for 3D reconstruction of cultural and natural history objects. So as we're digitizing these objects, you take a whole bunch of two-dimensional photographs of a particular object, and this is another dog skull here, and stitch them all together so that you can get a fully rendered three-dimensional model. And so as we put together these three-dimensional reconstructions of that, you can actually get from a series of anywhere from 200 to 300 pictures a fully realized three-dimensional model that can be 3D printed or shared in a context that allows you to zoom in and manipulate the model and to do serial slices in a virtual context. So that way the object is no longer just a dog skull on a shelf in the lab in the basement of the student building. This is a contextual contextually enhanced collection object that allows for us to tell us a whole lot more than just that story. And so as we're starting to think about ways in which photogrammetry can really enhance the utility of these collections, it is sort of a low-cost, low-entry barrier manner in which the digitization of specimens, as opposed to costly laser scanners or other expensive software, can allow for these sorts of things. But there's still a significant amount of computational resources that go into these reconstructions. And then in the IU Paleontology collection, we've got a whole bunch of cabinets in our newly renovated space, and so we frequently get these questions from students or from kids that come to visit for IU Science Fest or various things. So researchers frequently are asking about our conodonts from Carl Rexroth, who is a very prolific member of the Indiana Geological Survey, who just passed about a week ago. Contributed tremendously to this collection, particularly because of these early vertebrate fossils, or microfossils, really are very useful for determining the age of the Earth and the age of rocks, in which their use and their utility is greatly expanded by having the ability to date the rocks themselves, but also the fossils that are very widespread and abundant, cosmopolitan and wide-ranging, but have very short lifespans in the geological record. So we can see how they spread out geographically and tell us that we've got sort of this lateral context of a similar age type of rock, and using the fossils are considerably more useful. And so the conodonts are quite frequently the most frequent requested specimens in our collection. But the T-Rexes that we have, they're all casts, and they're all in our teaching collection, and we don't really have a full one. We've just got a tooth and a claw, and that's about it. But it's not just 1.5 million fossils. There's a whole lot more to this renovated space that is 5 classrooms where the walls were knocked out, and it's not just a whole bunch of cabinets with drawers and rocks and crumbling papers and dusty specimens. There's a rich history of dozens of scientists from this university and beyond, graduate students, faculty members, research scientists from the survey, and others that have contributed their life's work to this thing. And so we have lots more metadata that go into all of this. And so we have built finding aids with the library so we can determine how we can appropriately address the information technology needs for constructing a research program about the resources that we have here at this university. So whether it's 35mm slides that were used in the teaching and lecturing of individual faculty members, or slides about the research subjects that then went into publications or unpublished works, all of these things are reposited within the IU Paleontology collection. And there's a tremendous amount of material that is just waiting for someone to look at it, and has not been receiving a whole lot of attention, because there's 500 million years worth of history here. This is the history of life on the Earth. And so really understanding how all of these things came to be, especially in this area of the Midwest, really allows for us to expand our scope and our understanding of what life was like when the dinosaurs were here, because we don't have any of those rocks in this age. All of the rocks in the Monroe County and Indiana area are far too old to have contained any dinosaurs. So if anyone tells you that they found a dinosaur in Indiana, that's a non-avian dinosaur, as Chris and I were having this conversation earlier. That dinosaurs that are classically thought of are not even possible to have been found in Indiana. And so, as Craig was alluding earlier, some of the institutional buy-in to this project for enabling discoverability and accessibility of content and the democratization of science, and making sure that these are made available to researchers not only within our institution, because many of my colleagues in anthropology, biology, and geological sciences, where I do have a little tendency to create some stir, they really sometimes have no recollection of or knowledge that these collections do exist, even within our own institution. But now, generally speaking, as Imago is a digital content repository that is being broadcast to the entire world, we can send out some of these images of those physical specimens, whether they be a 3D model or a scan of a PDF of a field notebook that was collected at the same time as those physical specimens. And this is one way in which we're allowing for the greater public and researchers alike to access these collection objects, to enhance the ways in which their own search criteria for building research programs are able to be built and enhanced. And it's really that metadata context that allows for us to grow into greater understanding of what the semantic web and linked data really could look like. This is a way in which we can have a physical object linked to a born digital object, linked to an ORCID for the researcher that worked on that project, for a DOI to the publication that summarized the findings about those specimens in a greater context. And then linking all of these data practices so that way when you're researching a particular paper, you don't just go and crawl through the bibliography and track down papers that titles sound interesting. You can actually go back and use an international geological specimen number or something like that, where you can actually find a trackable persistent data identifier for that particular specimen itself and track down other publications in which that specimen was used. So that way we can have this greater understanding of this web of knowledge that allows for data or genome sequences or for publications or for researchers to all be intrinsically linked to the content from which they're derived, and hence the reproducibility and the accessibility of scientific material to broad communities. And so ways in which we're doing that are to really take the data that are written on the specimen labels that are tucked away in those neat little acid-free boxes that all of our specimens are collected in. So this bright zone here that is the holotype was found here in Indiana, actually just outside of Richmond, Indiana. And a holotype is the specimen for which a species is named. And so the very first Monticuliflora indianensis is that particular specimen. The IU Paleontology Collection has well over 1,000 of these for which a specimen has been designated as the original, the archetypal species for which that is named. And so this research is something that must be preserved for science to be able to use any Linnaean taxonomy or cladistic taxonomy, where we have, say, a fundamental discrete unit for which our definitions of nomenclature are based. And so in all of these, we can create photographs and spreadsheets and relational databases of all of the metadata, but we're now also producing three-dimensional models of all of these collections. So that way, we can allow for researchers to interact with these specimens in a greater context so they can evaluate whether or not a visit to the IU Paleontology Collection on the 5th floor of the Geology Building on Indiana University at Bloomington's campus will be a worthwhile visit, or if they would be able to aggregate that data set and to determine, well, I need to get these 5 specimens, so maybe I'll request those as a loan from IU Bloomington. I'll get these 250 from the Smithsonian Institution and put all of those together and put together this large-scale data framework for understanding the history and evolution of this particular species. And so really it is in data aggregation that allows for us to really begin to conceptualize and visualize what big data research is all about. And so as the RDA, the Research Data Alliance, EarthCube, and other organizations are beginning to focus on the utility of shared standards and controlled vocabulary for describing what we know and how we know things about particular collection objects, iDigBio is an organization that's known as the Integrated Digitized Biocollections Framework, and they are a central hub for amalgamating all of this data, metadata, and image information. So they've got 73 million in count. I mean, that was as of this morning. It's probably up to 74 by now. But the media records that are associated with that, whether they be a three-dimensional model or a photograph or whatever, frankly, I'm not surprised that I'm a lot surprised that there are not more media records associated with specimen records. Common delimited text files are really easy to share and to publish to a whole bunch of data frameworks and ingesters and aggregators across the world, like the Global Biodiversity Information Facility. But making sure that there is a framework by which we can share the research products themselves, not just the information about those research products, is something that Imago is hoping to enhance the utility of, so that way we can make the number of media records more in parity with the actual number of specimens for which we have that information. And as we're going into a little bit more information about functional morphology and using big data to understand how and why organisms live the way they did, fossils that you find here in Indiana are very commonly brachiopods or echinoderms or bryozoans and coral life, like benthic, sort of bottom-dwelling marine organisms, pardon me, that lived on this sort of shallow oceans that covered Indiana about 300 million years ago or more. And so looking at the ways in which these brachiopods interacted with their environment and with each other as the very dominant sort of biota from this region, pardon me, just getting over a little bit of sickness, we can start to understand how simulated flow and computational dynamics. I've got some water.
Oh, you got water. Okay, good. Yeah, Casey.
All right. So we're in good shape here. Sorry, my Italian cadence is a little bit rough today. So as we're starting to look about simulated fluid dynamics and functional morphology as the ways in which the organism and the preserved parts of the organism interact with their environment, we start to understand a little bit more about how these critters would have fed, and by ingesting detrital matter and little organic bits and pieces from the water column into their gill system inside the organism. So the way in which sort of fluid dynamics portrays themselves as portrayed here is that sort of sediment stability and the manner in which the organism was oriented on the sort of sediment-water interface on the ocean floor is really important, because if they get flipped over, they're not really mobile critters. They can't flip themselves back over in order to get their feeding apparatus, their gate between their shells, back into the water flow that allows for them to feed. Similarly, they also need to make sure that there is a preferred orientation that allows for that current flow to get in between their shells, that allows for that particulate matter that they feed upon as detritivores to sort of enable their ecological and evolutionary success. So we're working on a number of products right now to three-dimensionally scan all of these organisms so we can start to understand the ways in which these brachiopods would interact with their environment and with feeding mechanisms, so we know a little bit about their ecology and their evolutionary history. And a number of recent studies have done a number of simulations in sort of this actualistic paleontology, where they've created sort of these plaster molds and plastic replicas of these specimens. And this is about as tech savvy as the field has really come in a lot of recent years. And this is actually a 2010 study here, that we can now three-dimensionally print these models and suspend them in a sediment flume study analysis, where we've got a rotational system that can sort of validate and quantify sediment transport along the ocean floor in a simulated context. And then we can also simulate a number of functional morphologies for the brachiopods themselves and the ways that they're constructed. And so we've had a number of students that have been working with us to 3D scan these bivalves and brachiopods and to understand the ways in which they all come together, and you can see a very large range of morphologies, and the shapes of these critters really is very important for understanding the way in which they interact with their environment. And it's that evolution of shape that is most interesting to me for my own research, but also for other projects like this, where we'll be going to Tanzania this summer to actually help another museum digitize their collections. We'll be teaching them how to use photogrammetry for the mass digitization and three-dimensional representation of our earliest hominid ancestors. So we've got early human evolution in the cradle of humanity in Tanzania and Olduvai Gorge, where we'll be trying to help to promote the accessibility and the research potential for our understanding of the earliest human evolution. And so collection-based research is really something that's available for not only for the democratization of science and for increasing the availability and utility of scientific research products such as the collection objects themselves and all of the associated metadata, but it's also a way to get kids and the future scientists and other generations excited about what it means to be a scientist and to ask questions and to really understand what that context is that goes into making something more than just a pretty object that sits on a shelf somewhere in a nice little divider box with a whole bunch of labels. And it's not just stamp collecting, it's a whole lot more to it. And so at the Science Fest last week and a number of other events, we've got people that are very interested in what 3D printing can be done to enhance the tactile nature of learning and understanding how we can see how these things are built, or to use citizen science and have students that come together and use some laptops that were provided by Rob Peng for getting students to actually digitize and type in the metadata from the specimen labels on the objects themselves, and to understand what is in the collections here at IU. And we've even got an app that we've worked on developing with the Advanced Visualization Lab, where we have a 3D collection viewer where we can see in augmented reality the ways in which these things come together. And so imagine this on a sort of a horizontal giant iPad interface where you can start to go through and browse this collection, and you can use various search parameters to understand all of the relationships between all of these pretty objects on the shelf, where you can touch Indiana and turn on that search filter and look at the things that are just from Indiana or from North America, or you can search geographically or taxonomically, searching by the availability of the families that are related, or temporally, because the fossil record is very diverse and we've got close to 500 million years worth of research on all of this. But the individual collection objects themselves, like this one, the direwolf will be able to pull up a three-dimensional model of the actual direwolf maxilla that was found here in Bloomington. So we have direwolves that are not just in the storyland fantasy of Game of Thrones and all. Now everybody thinks that direwolves are now the new hot thing, but they used to be the new hot thing 20,000 years ago in Bloomington. And so these collection objects allow us to put a better framework onto how all of these things come together. And as I was mentioning a little bit about citizen science and broadening our customer base and exciting people about what data we have available, is notesfromnature.org is now a framework where we're working on looking at the specimen metadata associated with each individual collection object, putting it into this framework, and now live is now our IU collection of Cambrian trilobites from yet another former state geologist and significant paleontologist here at IU, as we've got these collections of plaster molds of a range of morphologies of trilobites from all over the world. So Charles Dice went and collected a whole bunch of museum specimens from literally the best museums around the world, made a plaster mold of them, and then curated them into an entire cabinet of several thousand plaster molds. And so this was well before 3D printing or 3D scanning was a thing, but understanding the ways in which you can replicate the availability of specimen data is something that we're also trying to promote. And similarly, as we are engaging citizen science, all of the labels are being transcribed into this framework. And while I do recognize that OCR and handwritten text recognition are utilities that I'm exploring further with colleagues here in RT, we started out with this particular set because they were handwritten to try and learn about how we can teach stratigraphy and concepts of earth science and geology to a community that has not had a lot of exposure to the geological sciences. So it's been a fun challenge. But also, most of the metadata that in the IU Paleontology collection are handwritten on these catalog ledgers. They are ridiculously standardized and meticulous in nature. However, handwritten sheets of paper are very difficult to work with, and we've got 30,000 of them, but that's only 30,000 pages for 1.5 million collection objects. So we'll still need to go to the individual labels and work out a scheme in which we can digitize all those and put them into a usable framework. So that way we can put together a broader study for empirical evaluation of factors in morphometric variation, how the study of shape itself is a useful parameter for understanding the evolution of a group of rodents, such as what David Polly works with. Or for my own particular expertise and for my dissertation work, where I travel to museums all around the world to put together a large data set of tens of thousands of fossil specimens, where looking at very subtle changes in morphology can actually tell you a lot about their ecology and their ways in which they interact with other species, and finding ways in which you can develop a sort of a machine learning model for differentiating species relatedness and taxonomic differentiation. So like the ways in which we can say that, oh, this species down here in this corner, these are all one particular species A, these are species B, and trying to put an a priori model on the differentiation of species based only upon their shape. So it's a little bit more complex than high school geometry, but by fitting an equation to the actual shape of the critter itself, you have a segregated model that allows you for a lot of very, very powerful metrics for understanding taxonomy and ecology. And all of this is because of the cyber infrastructure that allows for it to happen. And so while we do rely quite a bit on the aggregation of data and digital data that allows for all this to come together, 3D printing and 3D scanning allows for a richer context for all of these things to come together. And so we are very much taking advantage of the infrastructure and the ecosystem for data analysis and refinement that is offered here at IU. And so whether it's using high performance computing resources on Karst or Quarry or other Exceed resources, using geospatial data, 3D printing, and a number of other units, we really do intersect with a whole lot of these units of RT and the broader intersection of cyber infrastructure across a very large platform. And it really is that the big data that are now being able to be answered in biology and biodiversity, informatics, anthropology, archaeology, paleontology, and all of these other ologies that come together really do rely very much upon the services that are provided here in UITS. And so I want to thank you for all of your collaborations, because I have worked with a lot of you, and as Franco said before, I will continue to be making requests of you because these things do add up quite quickly. And especially thank you to all of you for being here. This is really fun to see an excited and engaged group of people that are interested in how we can share these data and services and make them available to a broader audience.
Let's thank our speakers. Gary and I got our undergraduate degrees about 35 miles and 27 years apart from each other. When I was an undergraduate, the state-of-the-art in paleontological imaging was a microscope, a camera lucidator, a Koh-i-Noor repeatograph pen, vellum paper, a student, and large quantities of Mountain Dew. It was a slow process, and I did a little teeny bit of it. And it's just really amazing to think how much more accessible the data in the form of the specimens and metadata that we have in the collections here are, how much more available they are now as compared to state-of-the-art when all the stuff was done by hand. So I'm really, really impressed and just amazed by the quality of the work. And I should also say that thanks to David Polly and Gary and some other folks, Indiana University is a hotbed of computational experimental paleontology. It's a, you know, what is being done in paleontological research with our computers here on top of just, you know, on top of the great work managing the specimens. It's really, really impressive. So with that comment, yeah, so I wasn't smart enough to be a geologist, but I always sort of wished I was. It's just neat science. Are there any questions?
Okay, when you talked about aggregation, you had a slide with a number of different organizations. How thick is that aggregation? Is there a standard metadata format that's getting passed back and forth? Are there APIs? You know, how deep does that really go?
One of the communities in which I am now very involved is working with the group called ePanda, which is Enhancing Paleontological and Neontological Data Discovery API. So that's the A on there is actually another acronym in itself. So APIs are sort of the translation between programming interfaces that allow for a derivation of the Dublin core metadata schema that you're very familiar with, which is what the biological community uses is now the Darwin core metadata schema. So for biological nomenclature and for a lot of the contextual information that goes into our understanding and cataloging of things, we use standardized vocabulary as best we can using the Darwin core metadata schema to, so the ways in which we use our cataloging and collection management software to then aggregate out to other aggregators through via an API is by you taking that standardized schema, distilling it into a standardized format, and then packaging that archive of spreadsheets basically and links to other resources. So having persistent digital identifiers like URLs and other things are quite useful for linking everything back together and then sending it back out. So really having those ontologies and metadata schemas worked out is the only way in which this is occurring.
Other questions?
Who do you work with for the photogrammetry that you talked about? Is that something that you do personally, or? It
is something that I do personally. There are a number of students that are working with us right now as well. It's something that is really, really taking off in terms of popularity on this campus. Matt Brennan and Bernie Fisher in Informatics are doing a lot of really high-quality photogrammetry work. Bill Sherman and others in the AVL are doing some photogrammetry work. Chris is also involved with that a little bit.
I want to do a brain.
You want to do a brain? Well, inside or outside?
Let's talk later.
Any other questions? Well, let's thank Gary and Franco again for two really wonderful talks.